So on Monday, we're going to finish up the Rubisco lab. We're going to run the gel, and we're going to stain the gel. You won't have the results of the gel that day because they really need to de-stain overnight. So I will image them for you on Tuesday and email them to you, and then you can start doing the analysis, which really isn't too involved. It's going to be very involved in the next lab that we do, which is Western blotting. But you'll still want to analyze it. I think that lab worked out pretty good. You guys did very well the other day, so good job. It's a tough lab. Be sure that you are able to do those calculations, right? So to get from a concentration of protein, determine it based on any dilution factors that were applied. Whether you multiply or divide, right? If you're using a 2x buffer, you have to multiply. If you diluted the protein before you read it on the spec, you have to divide. I'm sorry, other way around. You read it on the spec, dilute it before you do the spec, you have to multiply. If you add a 2x Lemley buffer, you have to divide. Okay, so we're going to shift gears, and like I said, today we're going to have a little light day. We're just going to do chapter 7. And the end of chapter 7, it's a big chapter, but so it talks about transcriptional regulation, which is really important in more complex cells like eukaryotic cells. And the last section of the chapter, I really we're just looking at a summary of the last section on epigenetics. And I want you to understand the principles of epigenetics, but I'm not going to require that you understand the specific mechanisms that are laid out in the textbook with specific examples. All right, so section 7.1 looks at transcriptional regulation in E. coli, and some of this should be a review for you from Bio 1. We're talking about the lactose operon as a real prototypical example, classical example of gene regulation. But we're going to apply a little twist to it that maybe you didn't cover in Bio 1, having to do with positive control of the operon. What's this? Okay, so what is an operon? Well, an operon is, it makes studies of prokaryotic genomics very nice because it really organizes the genes within that one chromosome that is found in bacteria. So operons are only present in bacteria, and it's an organizational step that has evolved, generated these clusters of genes that are typically associated with the same function. Okay? So an operon is a collection of genes on the bacterial chromosome that typically all have to do with whatever function. In the terms of lactose, it has to do with lactose metabolism. So the lactose mechanism, the mechanism of gene regulation, is really important for an important example of how a cell regulates its genetic activity to maximize efficiency. So only in the presence of lactose will the enzymes that are required to metabolize lactose actually be on. And when lactose is not present, transcription for those metabolizing enzymes will be off. That saves the cell a lot of energy, right? If there's no lactose around, why create the enzymes that are needed to metabolize it? All right, so a little bit about lactose metabolism. Lactose, which is a milk sugar, gets broken down into two disaccharides, which are isomers galactose and glucose. Right? And like an operon is a collection of genes. It contains typically, so the generic makeup of an operon is a promoter, and then it's followed by structural genes. So promoter is regulating expression of this. If RNA polymerase and sigma factor bind to the promoter region, transcription will be on, and you will get expression of the structural genes, that's proteins. All right, the lac operon has some specific components. It has the promoter region, and then it has three structural genes. Right, so the genes are beta-galactosidase, permease, and acetyltransferase. So beta-galactosidase and permease are directly involved in the metabolism of lactose. Beta-galactosidase is the one that actually hydrolyzes lactose into glucose and galactose. Permease is a membrane transporter. It helps bring lactose into the cell. Transacetylase, there's still a lot of debate on the purpose of this enzyme. It's not directly essential for the metabolism of lactose, but it's still included within this operon. All right, so the promoter and those three structural genes are an essential component of this operon, but there are also two other parts. There is a operator sequence, which is found over here, found downstream of the promoter. And then there is a regulatory element called a repressor. And this is our lac operon. So over here is beta-gal, permease, and transacetylase. So the repressor and the operator are involved in the regulation of this operon, whether it's going to be turned on or turned off. All right, so the repressor over here actually has its own promoter. And basically, expression of the repressor creates a protein all the time. The protein is always produced. So there's always repressor present in an E. coli cell. However, the structural genes are not always turned on. Okay? The job of the repressor is to bind to the operator and block transcription. So under normal conditions, that's what's going on. Under normal conditions, which you would say lactose is not present, lac operon is off. But this is what's happening. The repressor protein binds to the operator. RNA polymerase comes in, binds to the promoter, but can't travel down the operon to transcribe the structural genes. However, when lactose is present, lactose will actually, a metabolite of it, allolactose, will bind to lactose, will bind to the repressor, and binding of lactose to the repressor produces a conformational change of that protein. So now the repressor cannot bind to the operator. That means the RNA polymerase, in the presence of lactose, able to bind to the promoter and drive transcription of those genes. Okay, so when lactose is absent, under normal conditions, right, the repressor is bound to the operator, it blocks RNA polymerase, and you don't get expression of the structural genes. But when lactose is present, it binds to the repressor protein, change in shape, a conformational change, and it can't bind to the operator. All right, if it can't bind to the operator, RNA polymerase is free to travel down the DNA and transcribe LAC-C, LAC-Y, and LAC-A to get enzymes produced. Now,
is this how they make those biological logic gates?
Logic gates?
Yeah, supposedly.
So I wonder if, I haven't heard that term before, but I wonder if what you're talking about is the whole thing with beta-gal and cloning. So this mechanism is heavily exploited in molecular biology for cloning mechanisms. And we're going to do it later on in the lab. We're going to do blue-white cloning. So only in the presence of an inducer is beta-galactose produced, and you can get a signal from that. I wonder if that's what you're talking about.
I think it's in synthetic biology. Signal. The professors basically when the is on
It may very well be. I don't know. It's a good question. But this is heavily understood and heavily exploited. So it's used for all sorts of things. So it's probably used for that. Okay, so here's your summary slide of the mechanism of the lac operon. Okay, so here you've got RNA polymerase and sigma factor up there trying to bind to the promoter, but it can't because the repressor protein is bound to the operator. Right, so it's blocked, so you don't get transcription of those genes, and that's when lactose is not present. However, when lactose is present, it's bound to the repressor protein, changing its shape. Repressor protein can't bind to the operator, and so RNA polymerase and sigma factor can bind to the promoter and drive transcription of the structural genes. Not too bad. However, there is other signaling that is involved in the lac operon. The cell, the E. coli cell, has an ultimate preference for glucose, actually. Okay, so, if glucose is present, it doesn't need to metabolize lactose. So glucose is essentially a way of positive control of this system, whereas using the repressor protein and lactose, this is a form of negative control. All right, so in order to understand this, you have to understand the pathway that's involved in glucose metabolism in E. coli. So glucose is broken down and comes into the cell into an intermediate molecule called alpha-ketoglutarate. And alpha-ketoglutarate is a molecule that will inhibit a pathway that activates the promoter for transcription within the lac operon. So there's a molecule called adenylyl cyclase. Adenylyl cyclase converts adenine triphosphate, ATP, into cyclic AMP. We're going to look, so as we get into signaling pathways, you're going to start to see cyclic AMP is a very prominent molecule. It's very important. Right, so the adenylyl cyclase converts ATP into CAMP. Cyclic AMP activates this protein called catalyte activator protein, metabolite activator protein, or CAP. So when CAMP is bound to CAP, it's in its active form, and when it's in its active form, it recruits RNA polymerase to the promoter. Okay, so think about this. This is an inhibitory pathway, right? So if glucose levels are high, you get a lot of inhibition of this. This is all shut off. If glucose levels are low, though, then this pathway goes forward. All right, and that's the important caveat here. So because of that, the lac operon is really only strongly activated when both lactose is present and glucose is absent. Right? If lactose is present and glucose is absent, if lactose is present and glucose is present, then this pathway is off. You don't get expression of this. So the presence of glucose, or the lack of it, is something that can override the lac operon. All right, so with low glucose, you have high levels of CAMP because alpha-ketoglutarate is inhibited. And if you have high glucose, you have low levels of CMP. Right? When you have low glucose, the lac operon is on. When you have high glucose, the lactose operon is off. All right, so there's 4 possible scenarios in this example of gene regulation. And they're actually better summarized here in this figure. All right, you can have lactose and glucose absent. You would have no transcription, right? Because lactose is absent, the repressor protein is bound to the operator. With glucose being absent, though, catabolite activator protein is active, but RNA polymerase cannot get to the promoter because the repressor protein is already blocking it. When lactose is present and glucose is present, and so both are present, the repressor protein is inhibited. Right? It can't bind to the operator, but cap protein is also inhibited. It's inactive. So it can't bring RNA polymerase to the promoter. When lactose is absent and glucose is present, right? Because lactose is absent, you have a repressor protein bound to the operator region. And because glucose is present, cap protein is inactive. So the only situation where this operon is actually on is when the repressor is inactivated by lactose. So lactose is present. And when glucose is not around, so that catabolite activator protein is activated by CAMP, and RNA polymerase can actually get to the DNA. Right, so this is an important scenario here. You should understand the mechanisms going behind it. I expect a number of questions on the exam related to this mechanism. Okay. Are any questions about this mechanism? It's not too bad. All right. So again, in Bio 1, I think you covered the lac operon. Maybe you did or did not cover the pathway with glucose metabolism. Remember, E. coli prefers glucose over lactose. All right, so that is the classical example of transcriptional regulation in E. coli. And in bacterial systems, it's a very nice organized system where you have these operons set up and all the genes are, they're very close to each other in terms of locus on the chromosome. But in eukaryotes, it's a lot messier. It's very different. And that's what section 7.2 is going to cover. So transcription factors in eukaryotes. So it's the abundance and collective activity of transcription factors that guides transcriptional regulation in eukaryotic cells. With this myriad of transcription factors, you can get a more diverse and selective level of transcription. All right, so in the last chapter, we talked about the promoter region, but there are a number of other, what we're going to term cis-acting regulatory sequences that are found around the promoter that assist in the transcription of eukaryotic genes. So, and this is an important term. So, because you have cis-acting regulatory components and you also have trans-acting regulatory components. Both are prominent in eukaryotic systems. Cis-acting are very close to the target gene on the same chromosome. Trans-acting are far away from the target gene and usually found on different chromosomes. So, could you have trans-acting regulatory sequences in prokaryotes, for example? Why not? Okay. Is one chromosome. Yeah, so you don't have more than one chromosome in bacteria. So all regulatory elements are considered cis. So how would one identify putative sequences? So these are sequences that we assume to be regulatory. Right? So how do we identify these cis regulatory sequences? And one way to do that is to use a reporter system. Right? The most common ones utilize luciferase. Luciferase is an enzyme that produces light. It's found in fireflies. And you could actually take the sequence that you think is regulatory, clone it into a plasmid that has the luciferase system, and then test it in living cells to see, are there transcription factors or molecules that bind to that regulatory sequence. All right, so here's our reporter plasmid over here, and we've put into it some putative regulatory sequence. So it's some sequence that we think maybe transcription factors bind to and enhance transcription. And the gene that the transcription factors are going to turn on is the gene for luciferase. So when that gene gets transcribed, that cell is going to light up green, bright green. All right, so we take that plasmid with our putative sequence and a luciferase gene, and we transfect them into cells. And if there are elements in the cells, like transcription factors that bind to the regulatory sequence, the cells will glow. All right, so this is a real easy assay to do. You don't have to even use a microscope. You could just take the cells and put them into a luminometer. It's a lot cheaper than a fluorescent scope. And you can detect that signal because the cells glow so brightly. It's a very strong signal. All right, so more on the cis-acting regulatory sequences. The goal of all these sequences is going to be to enhance transcription. And we've talked about some of them already, the TATE box, the TATA box, the GC boxes, the -35 and -10 elements, the CCAAT box, all of these components, the BRE element. So all these things that different transcription factors like 2F2D bind to are considered cis regulatory elements, and they're actually part of that core promoter complex. A good example of a promoter that's actually found in herpes virus, but that's okay because a lot of eukaryotic promoters, in terms of evolution, actually come from viral integration, is the herpes simplex thymidine kinase gene. So if you look at this promoter sequence, it's got all these different sequences, -25, -50, -75. Here's +1, so that's where transcription begins. And in order to get good, efficient transcription of that gene, whatever it is, all of these different elements have to have a different transcription factor bound to them. All right, so those are sequences found at the promoter region. But in eukaryotes, there are also sequences that are found away from the promoter region, still considered cis regulatory sequences because they're on the same chromosome, that greatly increase transcriptional efficiency. And we call these enhancer sequences, right? And there's a lot of diversity and variability in these enhancer sequences in terms of where they can be located, whether they're located upstream of the promoter, downstream of the promoter, if they're in the forward or reverse orientation. That doesn't seem to matter much. But in order to get heightened transcription of genes in eukaryotes, you have to have these enhancer sequences. Right, so the SV40 enhancer is a good example of that. So here's your promoter. It's got the core regulatory sequences inside, the TATA box, you got your GC boxes. And then, so this intron, right, this can be really far away down the chromosome. You have this enhancer region. And the enhancer region is necessary to bind to transcription factors. What's going to happen is the DNA is actually going to loop, and the transcription factors that are bound to the enhancer are going to bind to that mediator protein that we talked about when we were talking about eukaryotic transcription. Remember, mediator protein is one of the last things that comes in after the basal transcription factors work. All right, so without enhancers and the transcription factors that are bound to them, you only get what's called a very basal level of transcription. And that just means as low as it can go and as normal as the level needs to be, usually to maintain some cellular homeostasis. But stimulation through enhancer sequences and the transcription factors that bind to them greatly increase the level of transcription. So look at this figure, important figure here. You can see without enhancers, as in A, you just get a basal level of transcription. You get a few mRNAs that are produced, ultimately low level of this protein of interest. All right. However, if it's a protein that the cell needs a lot of, it's going to require the use of enhancers to enhance transcription of that gene. The enhancer can be in close proximity to the gene. It can be far away from the promoter gene complex. It could be upstream or downstream of the promoter region. Or it can actually be in the reverse orientation. It doesn't matter. So it can be on the homologous chromosome. The reason, so when they found that, all this flexibility in like where the enhancer sequence can be located and the orientation that it needs or doesn't need to be in, started to indicate that maybe the DNA is looping around. And this is an important component not only of transcriptional regulation in eukaryotes, but also how the nucleus is organized in eukaryotic cells. Whoops. Right, so the purpose of this DNA looping is ultimately to organize the transcription of certain genes. Right? It does it in like a spatial sense. Right? The loop act with RNA polymerase at the promoter. Right? So, when you have your enhancer regulation involved, you have mediator complex that's coming in, and then you have the looping of the DNA to increase the activity of mediator and RNA polymerase. You get very precise control with how the DNA is looped and organized. So here you've got, this is just one chromosome, and it's kind of dated off here, but it's looping around like this. This is the promoter region over here. And the enhancer sequence is up here, and you've got some transcription factor or multiple transcription factors that are bound to that enhancer. The DNA is looped around, and it's held together by these particular protein called cohesion. And the cohesion is recruited to the DNA by these other DNA binding proteins. Right, but here you can see the looping. The specific transcription factor is interacting with mediator protein. And this is a good signal for RNA polymerase to get released. And it more efficiently regulates transcription, so you get a higher level of it. Okay, so how does the enhancer function? It binds to transcription factors. The enhancer may have several, usually does, have several sequence elements that bind to several transcription factors. Typically, the more transcription factors you have in an enhancer, the higher the level of expression. Right? So it's like a combined effect of all these different transcription factors coming together and converging on mediator protein at the promoter. So the Ig enhancer, is a good example of an enhancer sequence. When immunoglobulins are antibodies, right? So when an immune cell and a plasma B cell wants to produce antibody, it wants to produce antibody. Like that's its role. If it's got an antibody figured out and how to make it, it wants to make a lot of it. So the Ig enhancer is a very effective enhancer at driving up transcription of that particular antibody. Look at all of the different elements that are found within the Ig enhancer sequence. Right? For each one of those, it's a different transcription factor that binds to it, and collectively, all of those transcription factors produce a very heavy level of gene production. Okay, so because enhancer sequences they function through the looping of DNA, what we've noticed is the way the nucleus is actually organized. So this is the nucleus. There's protein on the inside of the membrane called lamina. And when the chromosomes, the way they're situated is they actually loop out like this. And we call each of these where they're looping a topologically associated domain, or TAD. And we know this because we find heavy amounts of mediator protein, which are really large proteins in the nucleus, all kind of localized like this. So within a topologically associated domain, you may have transcription of a collection of genes that typically are all involved in some pathway. So like prokaryotic systems have operons to organize genes, what research has found in the last decade is that these TADs, TA domains, TADs, are kind of the cell's way of organizing these functions as well. Okay. So in order to produce the looping, we talked about that cohesion protein, but there's also another important one, CTCF, which is a DNA binding protein. And it brings the two strands of DNA together so that cohesion can wrap around it and hold the loop together. All right, so these tabs are big, like it's extending out into the center of the nucleus, 100 kb. It's a pretty large loop. It's a 1 megabase, which is big. All right, so the reason why the CTCF regions are able to bring the DNA together is because they're very GC rich, right? And that cohesion is like a protein clamp that holds the ring together, okay? So here's your chromosome. This is like towards the periphery of the nucleus here. This is towards the center of the nucleus. And you can see the DNA looping out, cohesion ring holding these loops together. And it's, you can have enhancer sequences all throughout this region of DNA that is exacerbating or exciting mediator protein, right, to drive transcription of whatever your target gene is. All right, so TADs organize transcriptional control. Right, mediator complex is like a bridge between the multiple transcription factors that might be involved in binding to the enhancer sequences to ultimately bring RNA polymerase to the promoter region. Right? Spatial precision is what this is doing. Right? It's bringing all these complexes together. All right, so the last part of this section looks at two procedures that can be done in the lab. One called a electromobility shift assay, or EMSA, and the other one is called CHIP, which is stands for chromatin precipitation. So both of these have to do with recognizing regulatory sequences, right, and also recognizing transcription factors. Basically, these are the techniques that are used in the lab to identify which transcription factors, which remember are proteins, keep that in mind, are binding to regulatory elements, which are sequences on the DNA. Okay? All transcription factors are proteins. All right, so EMSA is similar to acrylamide gel electrophoresis, and we've covered that. So you understand how proteins can migrate through an acrylamide gel, right? They're coated with SDS. The larger proteins travel more slowly than the smaller ones. And EMSA is a bigger modification of that. So we're not only separating out proteins in the gel, but we're also applying to the gel pieces of DNA that are radio-labeled. So they have a radioactive tag on it. And then we can detect those. All right, so let's look at how this method works. So you take a DNA fragment, all right, a fragment that you think or that some protein is binding to a regulatory element. So some transcription factor is binding to, and you run it alone. Right, so that free DNA, you're going to be able to pick that up because it's radio-labeled. It's basically transfer all this stuff onto a membrane, with some X-ray film over it, and you can detect that signal right there. All right, another sample that you have is the same radio-labeled DNA, but you add a protein lysate to it, right? Or you can add recombinant protein, doesn't matter. You can add a single protein or thousands of proteins if you want to find out the protein that is binding to the element. So if a protein or a transcription factor binds to that DNA sequence, it's going to retard the migration, right? So you're going to get, you won't get all the DNA bound to the protein, so you'll still get a signal. But when DNA has that protein bound, it's going to migrate more slowly. So you're going to get another band that's up here. This, in EMSA vernacular, this is called a shift, because you have a transcription factor bound to the element. Now, if you have a guess on what protein it may be, and you have an antibody against that that transcription factor you can mix in with your lysate and with your DNA, an antibody specific against the transcription factor. And if it is in fact the correct antibody, then the antibody will bind to the protein, which will be bound to the DNA, which is a larger molecule collectively and migrates even more slowly through the gel. So in that case, you get what's called a super shift. Still getting the DNA, it's not bound, but now you get DNA plus antibody plus transcription factor, and it migrates more slowly, so you get a higher molecular weight band. So when you analyze this, this is like a PhD level procedure because you have to work with radiation. But when you analyze this stuff, you're analyzing two things. Like you're looking for a radioactive signal to get on X-ray film, but you're also looking for a signal from your antibody. So you have to actually do two separate gels at the same time. Okay, so that's EMSA, right? So EMSA is testing if a known transcription factor, right, because you're using an antibody against it, binds to a particular sequence. Okay, so all right, and so we'll talk a little bit about sequences, and then we'll talk about chromatin immunoprecipitation, and we're almost done. All right, so a little bit about these binding sequences. Where are these binding sequences found, first of all? So what are we talking about here? Transcription factor binding sites, where are they found? So they're found in the core promoter complex, and they're also found, what is it? Well, RNA polymerase is binding to these sequences. But where are these sequences found in the DNA? They're in the promoter region, I just said that, the core promoter region. Where else are they found on the DNA? Enhancers, very good. Right, that's where these sequences are found. So just a little bit about these sequences. They are consensus sequences, right? So consensus sequences are, like if you have a bunch of different consensus sequences and you look at them carefully, what you're going to notice is there are a lot of similarities within those different sequences. And that's why we call it a consensus sequence, because it has particular bases at particular positions that are very common among all of those sequences. Right? So there is flexibility in the base pairing. And it's this flexibility that permits a wide range of different transcription factors to come in. You think about the complexity of this, and this is what nature has basically provided over millions of years of evolution to get very complex living systems. So one way of kind of demonstrating and showing these consensus sequences is using it's called a position base matrix or position weight matrix. So here you've got a consensus sequence, CC, for example, one of them are CCAT, right? Now this specific example looks at serum response factor, or SRF. So serum response factor is a transcription factor in blood that binds to different serum response elements. And when it binds to those regulatory sequences, it drives transcription of genes that are involved in cell growth and cell differentiation. Okay? If you look at all these different SRE, right, serum recognition elements that are present in different genes, right, there are commonalities among the sequences. So the consensus sequence is CC, and then you have 6 bases that can be either A or T, followed by a GG, right? So all those SRE sequences have CC and GG at the beginning, and then there's flexibility on A's and T's in the middle. And what's nice about the position weight matrix is you can just quickly see like what is the preferential nucleotide at a particular position. So, in order to have this, you have to have this, and then there's flexibility within this region as well. All right, we're going to talk a lot. Yes, ma'am.
We don't have to have this short memorized or anything, right? But just understand how it
No,
works.
just understand how it works. Yeah. All right, so you'll be given some of these sequences maybe to analyze and whatnot. Okay, so all right, so again, the EMSA, right? Testing if a known transcription factor binds to a sequence. So it's really studying the sequence. It's looking at consensus sequences. Right. The other technique is called chromatin immunoprecipitation. And we've talked about immunoprecipitation. There was a question on your first exam, I think, about it. And so if you're immunoprecipitating something, you are using antibodies to bring down a particular protein of interest. Okay? And if we're looking at chromatin, well, obviously we're looking at, the nucleus, we're looking at the DNA, the genome. All right, so what are the steps in doing a chip? So the first thing is to treat the cell with formaldehyde. So formaldehyde is a fixative. And what it does is it freezes the cell in time. Proteins that are bound to structures will remain bound to them. The cell is no longer living. You're not losing any molecules through degradation. And it does this by producing cross-links. And that's why formaldehyde is such an efficient preservative and also why it's so toxic. So it permanently links DNA to proteins. So any proteins, any transcription factors that are bound to DNA are going to stay locked onto the DNA and bound. All right, so the next thing you do is you sonicate your chromatin, right? You don't want large chromosomes to work with. Instead, you want to blast those things up into little pieces, right? Sonication uses high-frequency sound waves to break up DNA. Then you apply an antibody against a specific transcription factor, right? And it will pull down the transcription factor that you're looking at. Because the transcription factor is cross-linked to the regulatory sequence, it will also pull down the regulatory sequence. All right, so from there, you can then dissociate the DNA from the transcription factor. That's by reversing the cross-links. You can do that. And you can purify the DNA, and then you can identify the consensus sequences either through PCR or through DNA sequencing. All right, so CHIP is a technique that reveals where transcription factors bind within the genome. So here you've got, and what's better about this system is it's a little closer to in vivo because you're actually, looking at live cells at one point, whereas with EMSA, it's more of a lysate that you're applying. Right? And that's important because, transcription factors bind to their elements depending on the conditions of the cell. Right? So as long as the cell conditions are optimal, it's more likely to bind. Trying to recapitulate that in the lab is difficult. All right, so you've got your cells and you treat them with formaldehyde, so everything sticks. Right? Any transcription factors bound to DNA will stay stuck to that DNA. You fragment the chromatin with sonication, and then you apply to the slurry of chopped up DNA your antibody that will bind to that specific transcription factor. And now you can immunoprecipitate it. You can pull it down because it's a heavier complex, and you can pull it out. So there's a way that you can purify that easily using beads that attract to the antibody. You can reverse the cross-links. So now we only have our sequence that was bound to the transcription factor that we're interested in, and we can identify the sequence now. That's one way that you can look at various consensus sequences. All right, so when you're comparing these two, EMSA is a little, they're both in vitro, right? Because they're not in an organism, but EMSA is more in vitro, more synthetic. It detects the DNA and protein binding using the electromobility shift, right? So when an antibody is bound to DNA, you get a shift. when protein is bound to DNA, you get a shift. When antibody and protein are bound to DNA, you get a super shift. With CHIP, though, you're looking at living cells, right? And you're pulling down specific transcription factors that are bound to it. So EMSIS asks the question of, does this protein bind to a DNA sequence? Whereas CHIP ultimately asks the question is, where in the genome, what specific sequence does this transcription factor bind to? So, there's a little overlap with the two techniques, but they're subtly different. And be sure you understand those differences, okay? All right, the last part of this section looks at transcription factors, and there are up to 20,000 of them in the cell. We're not going to really look at specific examples, but there is one way that you can kind of categorize them, and that's according to whether they are activators of transcription or if they are repressors of transcription. All right, transcriptional activators, and think about these proteins and what they're going to do. They have to bind to DNA. They bind to DNA, then they're going to contain a DNA binding domain, right? This is the domain that actually fits onto the DNA and locks in. And we're going to talk about some of these sequences later on. If you take my molecular biology class, we'll talk a lot about those those domains within proteins that bind to regulatory sequences. All right, so they have a portion of the protein that binds to the DNA. Then they have an activator domain as well, something that binds to mediator protein or binds to RNA polymerase or binds to other transcription factors. But ultimately, these transcriptional activators promote the binding of RNA polymerase II to the promoter. All right, so transcriptional repressors are a little different. They just contain repressor domains. All right, so they can directly inhibit activators. They can also interact with mediator proteins, but ultimately they block or compete with other transcription factors. Let's look at some figures here that'll help. All right, so here's an activator. Let's say it's binding to some enhancer sequence, so it has a DNA binding domain. And the two ways that it can activate transcription are through interacting with mediator and other transcription factors, or through actually modifying the structure of chromatin. And that we're going to talk about in the last section, but it's another way of regulating transcription, is how the chromatin is organized, the nucleosomes, and how tightly it's packed. Right, so so far, we've only really talked about this pathway, but there is another pathway. All right, repressor proteins, a little different, right? They can block the binding of an activator, so that's like they're competing with the regulatory sequence that they're binding to. Or they can repress mediator, right, from activating RNA polymerase, essentially. All right, so those are the two classes of transcription factors. You have activators and repressors. All right, you guys take a couple minute break and take 2 or 3 minutes, and we got another class activity for you guys. So these activities, I think, as you can see, the grades on them were really high. They're meant to boost your grade up. So keep doing a good job of them. Go ahead and break up in your groups, though. We'll pass these out, and we will give you guys like a half hour to work on this today, okay? Yeah.
Great. But it was about a different thing. So we had to do.
Everyone in the group should have the same grade. Well, something's wrong with that. So just come up after we get this going. Come up and show me what happened. I might have just put it in wrong.